Breakthrough in wheat resistance. First genetic map unveiled. Breaking. Chinese researchers have achieved a milestone that could revolutionize how we combat one of the world's most devastating crop diseases. In what may be described as a historic breakthrough, scientists from the Northwest Agriculture and Forestry University and the Chinese Academy of Sciences Institute of Genetics and Developmental Biology have introduced the very first genetic map that tracks wheat resistance to yellow rust, a pathogen infamously dubbed wheat cancer. Published in the esteemed journal Nature Genetics on July 23rd, this pioneering study offers plant breeders an extraordinary new tool, a comprehensive gene navigation system. This resource is set to enable the development of wheat varieties that can resist the ravages of yellow rust, thereby prolonging the effective life of these crops and reducing the heavy reliance on pesticides. But here's where it gets controversial. The widespread application of these genes might shift the dynamics of agricultural genetics in ways that some experts believe could change global breeding practices for decades. Yellow rust, caused by the rapidly evolving pathogen Puccinia strivormis fsp twitigi, PST, is notorious for its ability to mutate and replace prominent strains every 5 years, resulting in an alarming annual loss of about 10% of global wheat production. As Professor Kem Jincheng of the Chinese Academy of Engineering and leader of NWAFU's plant immunity team explains, cultivating resistant wheat varieties is absolutely critical to halting this recurring devastation. This study is the first of its kind to systematically document the genomic selection signatures and the intricate epidemiological interplay between wheat and PST over the past century. It also delves into the co-evolutionary race between resistance genes and newly emerging pathogen races. Over the course of 5 intense years, the team examined variome data from 2,191 wheat accessions spanning the globe and meticulously analyzed over 47,000 yellow rust response records across various environments and pathogen races. This rigorous process led to the identification of 431 resistance loci, allowing researchers to construct a genome-wide map that detailed where yellow rust resistance genes are located. The study didn't stop there. By closely investigating 559 candidate genes related to yellow rust resistance, the team successfully cloned 3 novel resistance genes linked to specific loci. One particularly exciting discovery is the gene known as YR5X. This new allele exhibits resistance to several PST races, underscoring the importance of allelic variation in diversifying resistance strategies. Meanwhile, gene YR6PM5 has shown the unique ability to fend off both yellow rust and the fungus responsible for powdery mildew, Plumeria graminis fsp draki, suggesting an innovative mechanism for conferring broad-spectrum disease resistance in crops. Additionally, the gene YRKB, TAEDR2B, strikes a remarkable balance by offering broad-spectrum protection against rust without jeopardizing crop yield. The identification of these elite haplotypes, genes that offer potent resistance along with favorable agronomic traits, is hailed as a major step forward. Professor Han Dejun from NWAFU notes that this genetic map is a treasure trove for modern wheat breeding programs, potentially extending the resisted lifespan of wheat varieties well beyond the typical 3 to 5 years seen today. With wheat breeding lines carrying these newly cloned genes now undergoing field trials in key wheat producing areas, both domestically and internationally, the implications of this work for reducing pesticide use and enhancing global food security are enormous. And this is the part most people miss. Could these advancements also spark a debate about the balance between genetic modifications and natural evolution in agriculture? What are your thoughts on integrating such genetic breakthroughs into global food systems? Speak up in the comments. Unlocking wheat secrets. Genomics for yellow rust resistance. The exploration of genomics has led to the identification of exceptional alleles and genes that confer resistance to yellow rust in wheat, a significant threat to global food security. Data availability. The foundational sequence data for whole genome sequencing, GDS, and RNA sequencing, RNA-seq, presented in this study has been archived in the National Center for Biotechnology Information, NCBI, under project numbers PRJNA12539 and PRJNA1256661. These datasets are publicly accessible at the NCBI website. Additionally, the data has been stored in the Genome Sequence Archive at the National Genomics Data Center, which is part of the China National Center for Bioinformation and the Beijing Institute of Genomics, Chinese Academy of Sciences. The specific accessions include CRA 004322, CRA 014998, CRA 004878, and CRA 021484, all of which can be found on the NGDZ website. Furthermore, genotype data in Varia call format, VCF, can be accessed under the accession number GVM00830. This genotype data is also available through Zenodo. To streamline access and expedite the breeding process for stripe rust resistance, we have created a specialized database. All cloned genes, including PQ112656, YER5X, previously known as ER5C, PQ112657, ER6, and PQ112658, TAR-DIR2, are available in the NCBI GenBank. Source data accompanying this paper can be found in the provided references. Code availability. All software utilized in this research is publicly available online, as detailed in the methods section and the reporting summary. References. 1. FAO-Stat Crops and Livestock Products, Food and Agriculture Organization of the United Nations, HAO, access 12 January 2024. https://www.faostat-qc. 2. Savary et al. The Global Burden of Pathogens and Pests on Major Food Crops, Nat Eagle Level 3, 430-439, 2019. 3. Hafiz et al. Creation and Judicious Application of the Wheat Resistance Gene Atlas, Mull Plant 14, 1053, 1070, 2021. Hovmower, MS Walter, Access Can 2020. Escalating Threat of Wheat Rusts, Science 329, 369, 2010. Hovmuller, MS, FAC, T, and GIF in Sleason and Animal, and Siveril in the Context of Plant Health. Current Open Microbiome 71, 102243, 2023. Wellings, CR, Global Status of Stripe Rust, a review of historical and current threats. Euphytica 179, 129 to 141, 2011. 7. Chen X, Pathogens Which Threaten Food Security, Puccinia striforis, the Wheat Stripe Rust Pathogen. FoodSec 12, 239 to 251, 2020. 8. BetaJN L, Research Investment Implications of Shifts in the Global Geography of Wheat Stripe Rust, NatPlitz 1, 15332, 2015. Bouvet et al., The Evolving Battle Up Between Yellow Rust and Wheat, Implications for Global Food Security, Fiori Abo Jeanette, 135, 741 to 753, 2022. And then McIntosh R, Mu, J, Hundy, and Kang Z, Wheat Stripe Rust Resistance Gene, YR24, YR26, a retrospective review. Croc J, 6131 to 329, 2018. Acknowledgements. We would like to express our gratitude to Prof. Changfa Wang for his invaluable contributions to our resistance BD efforts. We also extend our thanks to the dedicated members of the Plant Immunity Research Team at the State Key Laboratory of Crop Stress Resistance and High Efficiency Production at Northwest A&F University for their insightful comments. Special thanks to M. Wang, Ji Li, W. Guo, Ji Li, and C. Jiang for their constructive suggestions and discussions. We appreciate R.A. McIntosh for his assistance in language editing and proofreading the manuscript. Additionally, we thank J. Guo, X. Bai, and X. Huang for their help with genetic transformation, and X. Huang, Hu Zhang, and X. Zhou for conducting RT-qPCR assays and AQP phenotyping. We are grateful to H. Sal and F. Yuan for their support in confocal experiments, and H. Wang for constructing the genomic DNA library. We also acknowledge J. Tong for sharing critical YR resistance QTL information, and J. Du, Y. Peng, L. Zhang, W. Zhang, Z. Shu, Yi Yang, B. Cheng, and Q. Yao for their assistance in phenotyping. This research was supported by the National Natural Science Foundation of China and various other grants themed at enhancing agricultural resilience. Chinese scientists crack wheat cancer code. Boost global food security with genetic breakthrough. In a groundbreaking study, Chinese researchers have successfully mapped the genetic resistance to a disease often referred to as wheat cancer. This significant research, published in the prestigious journal Nature Genetics, was a collaborative effort between the Northwest Agriculture and Forestry University, NWAFU, and the Institute of Genetics and Developmental Biology at the Chinese Academy of Sciences. The findings offer breeders an innovative tool for gene navigation, as reported by the university to Xinhua on Friday. Yellow rust, caused by the pathogen Puccinia striaformis F. sp. triticum, PST, is notorious for its rapid mutation rate, producing a new dominant pathotype approximately every 5 years. This relentless evolution results in an annual loss of about 10% of the global wheat yield, according to Kang Zhensheng, an esteemed academician at the Chinese Academy of Engineering and a leading professor in NWAFU's plant immunity team. He emphasizes the importance of developing resistant wheat cultivars to effectively combat this disease. Kang stated, "For the first time, this study systematically reveals the genomic selection signatures and epidemiological characteristics of wheat-PST interactions over the past century, as well as the co-evolutionary dynamics between resistance genes and pathogen races." The research team dedicated 5 years to a thorough analysis, utilizing viriome data from 2,191 wheat accessions worldwide, alongside over 47,000 records of yellow rust responses from various environments and pathogen races. This extensive data collection enabled them to identify 431 loci associated with yellow rust resistance, leading to the creation of a comprehensive genome-wide map of these resistance genes. Through meticulous examination of 559 candidate genes linked to yellow rust resistance, the team successfully cloned 3 novel resistance genes corresponding to the identified loci. One of these, gene YR5X, is a new allele that shows resistance to multiple PST races, indicating the potential for allelic variation to enhance resistance spectra. Another significant discovery is gene YAR6PM5, which provides dual resistance against both yellow rust and Plumeria graminis fsp triticum, the latter being a type of powdery mildew. This finding reveals a new mechanism for achieving broad-spectrum disease resistance in crops. Additionally, gene YRKB Ta EDR2B offers broad-spectrum rust resistance without compromising yield. The identification of these elite haplotypes, which combine resistance with favorable agronomic traits, marks a substantial advancement in the field, according to the research team. Han Dejun, a professor at NWAFU, remarked, "This map is a rich resource for resistance gene deployment in wheat breeding programs." He further noted that the combinations of resistance genes identified could significantly extend the effective lifespan of resistant wheat varieties. Unlike current varieties that typically lose their resistance within 3 to 5 years due to pathogen evolution, these new genes could maintain their effectiveness for over a decade and possibly even longer. Currently, wheat breeding lines that incorporate these clone genes are undergoing field trials in major wheat growing regions, both domestically and internationally. This progress not only provides strong scientific backing to reduce pesticide usage, but also aims to bolster global food security against this ongoing threat. In a related note, it was previously reported that Chinese scientists have developed an innovative marshmallow concrete designed to safely halt aircraft during emergency landings.